Bismillahirrahmanirrahim. Started with the name of Almighty Allah, the most beneficent, the most merciful. Very first class, I did talk to you what's medicinal chemistry, and I did mention to you medicinal chemistry is being taught by triangle, and that triangle constituted, here is the pharmacist in the middle of clinician and nurse. So this triangle is pretty good to maintain the health scenario. And in that respect, I did talk to you very firstly, how would you like to define the medicinal chemistry? I did talk to you, it's a branch of chemistry that deals with the biochemical pathways through which drug produces their beneficial effects. This was the most preliminary definition. And then I did mention to you the most important definition in that respect, which is being prevailing in the global village now currently in the present scenario. And I did talk to you in that respect. As defined by IUPAC system, medicinal chemistry is a chemistry-based discipline involving aspects of biological, medical, and pharmaceutical sciences. It's concerned with the invention, discovery, design, identification, and preparation of biologically active compounds, the study of their metabolism, the interpretation of their mode of action at the molecular level, and the construction of structure-activity relationship, which is the relationship between chemical structure and pharmacological activity for a series of compounds. And I did explain to you while I was taking class very firstly. Now the next talk which we are going to share, that is important in that respect, is as we look back 38 years to the first edition of the FOI's Principal Medicinal Chemistry, nearly 63 years, but I would like to say now, considering this, there should be 83 years to the first edition of Wilson and Gisworth textbook Organic Chemistry in Pharmacy, later renamed Textbook of Organic Medicinal and Pharmaceutical Chemistry. We can examine how the teaching of medicinal chemistry has evolved over the last half of the 20th century. 60 years ago, almost I should say now 83 years ago, the approach to teaching drug classification was based on chemical functional groups in 1970s. As you can see, I did talk to you, if this is the receptor, and you can see this is the drug is acting in this way, like this, and these are the functional groups according to the previous definition, which I am going to explain it to you. This is the receptor, and functional groups are being entangled and kept herein. So this is the way which was the previous definition, which is going to be explained in this way. Lock and key mechanism would be there if these are the functional group, and they are being kept up by the receptor, and when they fully packed herein, jam packed would be there, and then the drug response would be illicit, otherwise not. No, this is one functional group, this is another functional group, this is another functional group, and this is another functional group, and this is another functional group. Those functional groups have been entrapped in the receptor, and the drug response elicits. The approach to the drug classification was based on the chemical functional group. In 1970s, it was the relationship between chemical structure and pharmacological activity for a series of the compound, and today medicinal chemistry involves the integration of these principles with pharmacology, pharmaceutics, and therapeutics into a single multi-semester course called Pharmacodynamics, Pharmacotherapeutics, or other similar names. Drug discovery and development will always maintain its role in the traditional drug therapy, but its application to pharmacogenomics, this is the most important aspect which currently we have to share. An entire dilemma of the talk related to medicines are going on pharmacogenomics. These are the three words. If you could see, apparently it is a single word, but it comprises of three words. Pharmacogenetics. This is one word, this is another word, and there is co in between these two. So these two words are being correlated through co in it. So no, you can say pharma means drug, genomics means gene. Now you can say what drug do with the gene, and vice versa is also correct. What gene do with the drug? That is the dilemma of the talk of current century. And the scientists are exploring these things, what this science says. This is the most powerful science. What is genetics there and inside? And how does that altered genetics could be corrected by administering the pretty good drug in that aspect? And that's why this science is being correlated in this way or in that way. So pharmacogenomics is the actual dilemma of the talk of current century. May well become the treatment modality of future. In drug discovery, toxogenomic is used to improve the safety of the drug manufactured by US Food and Drug Administration. Now again here I would like to present you that is ADME was the dilemma of the talk. Now then T was also incorporated into it. ADME 1, 2, 3, 4 as the fingers of the hand forward. Now the fifth one is also included in it. 5 fingers have been completed: absorption, distribution, metabolism, excretion, and toxicology. That's why the word has been used, toxogenomic, which was not included previously, but now currently if you would see the latest books of pharmacists related to pharmacy, definitely the word T has been included. That indicates the toxicity, which is very, very important, and it is the best role of pharmacists to know everyone of individual existing in the global village. How does this toxicity is important? I'm strange. The toxicity is not the domain of the clinician. The clinician never goes to the toxicity. They only like to know secondary infection should not be there. Therefore, they keep on taking many drugs. But the pharmacist would like to take the drug when it is needed. And this is very, very necessary because the toxicity is more deleterious than administering the drug. So you should have to understand very safe and therapeutic, efficacious drug should be administered into the body, and that is the domain of a pharmacist, and pharmacist has to clarify these things to the clinician because they are more oriented towards drug, and you are the person who make the drug and you are more oriented in a way to clarify the clinician about the word toxicity, which is more important and very, very necessary, and this is the domain of a pharmacist. My dear son and daughter, could you listen to me? That's very, very important for you and you should know about it. Toxicogenomics is used to improve the safety of the drug mentored by US Food and Drug Administration FDA by studying the adverse bleak toxic effects of the drugs in order to draw conclusion on the toxic and safety risk to patient. That is more important. The scope of knowledge in organic chemistry, biochemistry, pharmacology and therapeutics allows students to make generalization connecting the pharmacochemical properties of small organic molecules and peptides to the receptor and biochemical properties of living system. Now, creating new drug to combat disease is a complex process Can you understand? Creating a new drugs to combat disease is a complex process. The shape of a drug must be right to allow it to bind to the specific disease-related protein that is called as a receptor. And already I did explain it to you in this way. That is, this is a receptor and it is protein in nature. If you could see, all protein is therein, which is responsible to constitute the receptor, and this is the drug. And now this drug receptor action should be there rightly. If it could be therein, then it's possible to elicit the response. Protein in nature, receptor, and this is the drug, and drug is administering into the receptor, and it is being entangled right in a fastening way, right, like this. Then the response would be there and otherwise not. For that matter, the drug shape, its presentation, its structure, its outer structure, its inner structure should be in a way that it should be able to catch up in the receptor, and if once it will be catch up in the receptor, drug response will be elicit as it is required. Receptor and to work effectively, this shape is determined by the core framework of molecule and the relative orientation of functional groups. Not that. This is the drug, and this area is the core work, this area, and these are the functional groups. When this core area has got these functional groups, and it can be easily catch up by the receptor. This lock and key mechanism is very, very important for a drug to produce its response. And this is the core study of the medicinal chemistry. If you could be knowing these things, then you will be able to understand. Now, the things are going on, latest module, latest modern presentation has been there, which will let you to explain this thing. Through receptor. Previously it was only the name receptor. Now many clear things are given herein. To work effectively, this shape is determined by the core framework of the molecule and the relative orientation of functional group in three-dimensional space. As a consequence, these generalizations validate by repetitive examples emerging in time as principles of drug discovery and drug mechanism, principles that describe the structural relationship between diverse organic molecule and the biomolecule functions that predict their mechanism towards controlling disease. Tahir is your domain, which is being mentioned here, in medicinal chemistry is central to modern drug discovery and development. Central to modern drug discovery, for most of the 20th century, the majority of the drugs were discovered either by identifying the active ingredients in traditional natural remedies, by rational drug design, or by serendipity. What do you mean by serendipity? This is the natural way, traditional way, as the people sitting on the roads are working as drug sellers. That means तुक्का, तीर तुक्के से पता कर लेना. यहाँ तो ये कि you goes on. Traditionally people could be able to know, but serendipity mean तुक्के से. वैसे you have administered a thing and you could be able to get a response. As we have moved into the 21st century, drug discovery has focused on drug targets and high throughput screening of the drug hits and computer assessed drug designed to fill its drug pipelines. Now you can understand, here is the traditional way as the hukama are doing, and you should be able to know by nature there are remedies in the plants and plants are being used, and then sometime by hit and trial method you could be able to know the, what you call it, serpentity. We have been moved 21st century. Drug discovery has focused on drug targets and high throughput screening of drug hits. So this is hit and trial method. What you call it as serendipity, tukke se, and computer assessed drug design. Now we are being, that tukka is being correlated towards the computer assessed drug design to fill its drug pipeline. Medicinal chemistry has advanced during the past several decades from not only synthesizing new compounds but to understanding the molecular basis of the disease and its control, identifying biomolecular targets implicated as disease causing and ultimately involving specific compound called hits. So this is the hit. That means by chance you could be able to get a thing and then you modify and then you get a lead. From the lead you could be able to have a series of the compound, as I did mention to you while I was taking the class very early, face to face. You can recall these things, and if you cannot, please do talk to me. And those who were not present in that time in the class, they cannot understand. And it is the duty of others to let them to know. If they even cannot, so this is again my prime duty to let them to explain, and don't hesitate to ask me in that respect. Called hits that block the biomolecular from progressing to illness or stop the disease into its tracks. Medicinal chemists use structure-activity relationship to improve the hits. Now, you got a compound by hit and trial, it was acting as a medicine and the effect was very feeble, and now you make series of compounds through structure-activity relationship, and you could be able to try each and individual compound in the body in vivo, and you could be able to get a lead which has got the most optimized biological activity which is needed. That is called as hit into lead candidates by optimizing their selectivity against the specific target and reducing drug activity against a non-target and ensuring appropriate pharmacokinetics properties involving drug distribution and clearance. Now again, pharmacokinetics is the more important. What is the rate constant in here while you are manufacturing, formulating the drug, and you could have to consider one compartment model system, two compartment model system, or somewhere three compartment model system. And the three compartment model system, you are very knowing where it is applied, where whosoever is very fatty body, that fatty body constitute the third open compartment. That means the fat is again a third compartment. In that respect, properties involving drug distribution and clearance. These are taught times for drug industry as companies are looking at diminishing pipeline of potential new drugs, growing competition from generic variations of their drugs, and increasing pressure from regulatory agencies to ensure that products are both safe and more efficacious. This is the last and least, wherein with Adami we have attached T, that is that products are both safe and more efficacious than existing drug. That is the domain wherein you should have to make a new medicine, and you can make in a way that if the newly made medicine should be more safer and more effective than the previous one. That is the dilemma and the talk of a pharmacist, and pharmacist has to generate more and more new and new biomolecules so as to combat the disease-oriented ailing humanities in the global village. With the completion of sequencing of the human genome, there are now greater challenges facing the drug industry for application of new technologies in discovery and development. Now you can see it was the Watson, great scientist. He could be able to give the human genome, and human genome comprises of almost 1 lakh gene. I did talk to you, and out of that 70,000 are effective and rest of the 30,000 genes are surplus in the body. So now the book is talking about the genomics. That means if you could be able to see where is gene, what gene is aberrant. Aberrant means जो के disorder हो गया है, that is called as aberrant gene. And if you replace those aberrant gene and in the place of that repaired gene has been placed, no problem at all. So if that is therein, you have not to administer the drug, and when that is not therein, you should have to administer the drug. But the most modern science, gene therapy, leads that the gene should be changed so that no need to give the drug to the patient. So this is the dilemma of the talk of currently which is going on in the global village, and it is the role of pharmacists to transport the gene where an aberrant gene is there. The number of drugs target once considered to be less than 500 has doubled, and it expected to increase tenfold. Look at how modern medicines are flourishing and coming. Diseases that were once thought to be caused by a single pathology are now known to have different etiologies requiring highly specific medications. Now you can understand this point. Disease that were once thought to be caused by a single pathology, means single gene was aberrant and disordered, and due to single gene disorder disease was there. But now to have different etiologies requiring high specific medicine, it is not necessarily that only the one gene is responsible. Now many genes are responsible to control one disease. For that matter, most modern medicines are there in order to maintain its medication. Now you can see there was previously, this was the only one gene which is responsible to produce the disease. Now I can understand this thing. I would like you to explain in this way, that is, if this is the DNA molecule and this is one gene, it is producing a disease. Now this means single pathology oriented problem, but now it's not the single gene. Now you can see this is liver cell, this is say a GIT cell, and this is say heart cell. Quite a few nucleotides will come from here, quite a few nucleotides will come from here, quite a few nucleotides come from here, and they will constitute a DNA molecule, and this DNA will produce. So this is multidisciplinary disease. Multidisciplinary disease. So multidisciplinary disease is there. So now single gene causing a disease, multiple gene causing a disease. I should not say multiple gene, but quite a few nucleotides are coming from here, quite a few nucleotides are coming from here, quite a few nucleotides are coming from here, and constituting a DNA, and they frame up a new gene, and that gene produces a disease, and you should have to control it. How you would be able to do? For that matter, we need a classical modern medicine through which you can control. Herein you can understand your domain, where you lies. And I know it, your domain is quite high up, higher and higher. And I used to say, तुंदी आवाज़ मुखालफ से ना घबरा ए उकाब, ये तो चलती है तुझे ऊंचा उड़ाने के लिए. पढ़ोगे, लिखोगे, बनोगे नवाब, खेलोगे, कूदोगे, होगे खराब. My son, you can say, quite modern medicine are being delivered to you by the grace of Almighty Allah. And I'm very happy at the moment that I am delivering this thing to you and you are trying to understand it. I used to say to you people, I can't let you to be less than any way from theory, but I cannot be you on the top of the global village in practicalities, because I myself is a hero. My country is zero in this respect, but its heroes are Jews sitting in the Utah University in America. But I would like you to say, you can go to America after completing your terminating studies of 5 years. Then I am producing raw material and finished goods would be manufactured there in America, China, UK, Italy, France, Germany, and so on and so forth, who are in a higher way in this aspect. The drug industry is integrating biopharmaceuticals such as pharmaceutical antibody. Now, this is the most important point. Disease that were once thought to be caused by a single pathology are now known to have different etiology, requiring highly specific medication in order to maintain its pipeline of new drugs. The drug industry is integrating, now you can understand this thing, integrating biopharmaceuticals such as therapeutics, antibodies. For example, in the treatment of arthritis, that again the credit goes towards monoclonal antibodies. And I would like to share the synthesis monoclonal antibodies while we're going a little bit ahead, along with smaller molecule drugs. As the drug industry undergoes reform, drug companies are developing collaborations with academia for new sources of drug molecules. Now, MOUs are being signed with professors, with various universities, professors in the global village, with the pharmaceuticals to manufacture new innovative drugs. That is why the word has been used, academia, for new sources of drug molecules. The editors of this textbook are all medicinal chemists, and our approach to editing this 7th edition of first principle of medicinal chemistry are influenced or respective academic backgrounds. Now, again, the academia, there have been professors, associate professors, assistant professors, lecturers residing in the universities background. We believe that our collaboration on this textbook represents a moleculing of our perspective that will provide new dimension of appreciation and understanding for all students. Now you can understand, my son and daughters, in nutshell, it is the medicinal chemist. You being a pharmacist is acting as a medicinal chemist, and you have to prepare the medicine. And in this way, I did mention to you, all medicinal chemists have written this book, and this is the specific book for you. And I did talk to you, even more specific, Berger is there, and even more specific towards chemistry related, Gisword is there. And these are the 3 books. And always I could have Foy in my hands, because this is between these 2 books, that is between Berger and Gisword. It gives the knowledge of biology as well, along with that chemistry. And Gisword is specific towards chemistry, and the Berger you cannot understand, its language is very high. So my son and daughter, thank you very much for listening to me, If you could not understand anything, you can talk to me. I would again like to provide you my mobile number 030041630075, as all of you could have this with you. Don't hesitate to ask me anything, anywhere, anytime, except while I'm sleeping, and that is the time from 11 p.m. to till 4:30 a.m. early in the morning. I am at your disposal. Thank you very much, my son and daughter. God bless you, which you blessed to be. Allah Hafiz, fi amanullah. Assalamu alaikum.